So please, let's go and support our students. So we have Mr. Podo, we have Christine Moore, and we have John Sandy, who are both presenting at the other side. So please go and give them all your support. So let's fill the rooms, please, and not be all, on one side. Otherwise, it will be imbalanced. Right. So our next session for the time being is still under the theme that we had earlier, and our chair is in the person of Dr. Aliu. So Dr. Aliu, if you're around, could you please take over from me and then chair the session? Thank you. And please be widely awake whilst we listen to the presentations. Thank you.
Thank you, Gloria. Thank you. I want to appreciate WANIDA for organizing this very memorable occasion. It's obviously a very huge success. Our next session will start with an online presentation, but it seems the presenter, is the presenter around? Okay, the online presentation has been shifted, so I want to see if the Professor Amidou Sami is around. Professor Amidou Sami is Associate Professor in the Department of Microbiology, University of Benda, South Africa. He's going to present on interleukin-10 production and seroprevalence of entamoeba histolytica infection among HIV-infected patients in South Africa. Professor Amidou, welcome.
Yeah, good afternoon, ladies and gentlemen. Welcome to this session. I would like to continue with, by saying congratulations to the WANIDA team for the brilliant organization, and also to say thank you for inviting us and giving us the opportunity, to share and to be able to create new links that might lead to further development for the young scientists in the continent. So I'm going to present a piece of work that was done by one of my PhD students, Dr. Ngobeni, who is now a postdoc in the US, on interleukin-10 production and seroprevalence of entamoeba histolytica. So as a guide of introduction, diarrheal diseases, as you know, is a very important cause of morbidity and mortality, particularly among children in many developing countries. And one of the causes of diarrheal diseases is entamoeba histolytica, which causes amoebiasis. This topic is very special because almost nobody has done studies on entamoeba histolytica since we started, the symposium. And it is, I believe it's an important cause of mortality in many countries. So if you look at the graph, you will see that diarrheal diseases represent 13%. 13% of all deaths, among children, followed by, AIDS, pneumonia, and then others, which are responsible for mortality among children less than 5 years old. And amoebiasis is the infection by entamoeba histolytica, and it's one of the most problematic because it has two different types of disease presentation, the intestinal and extraintestinal. Although the extraintestinal infections only constitute, less than 1%. So this is the life cycle of amoebiasis. So generally, either we can get the cyst from food, from our hands if they are dirty, or from water that we are drinking. So we get the cyst, and then those cysts are ingested. So once in the stomach, then they are existed, and then in the intestine, then they are going to cause the disease they can cause colitis, and then some of the entamoeba trophozoites are going to become cysts, and then will be excreted, with the stool samples. And those sometimes can find their ways into water systems, or can contaminate vegetables, or can be also transported by flies to the food that we are eating, and then we get contaminated again. So once entamoeba histolytica get into the stomach, so then they're going to produce enzymes, particularly cysteine proteinases, as well as many other enzymes that are going to participate in the degradation of the mucous layer on the intestine and facilitate the infection by entamoeba histolytica, which is going to produce those more cysteine that are going to degrade the wall, and then there will be activation of a certain number of cytokines, and we can see, for example, IL-1 beta, IL-1-6, IL-8, IL-12, interferon gamma, TNF alpha, and these, the product of these cytokines are going to create the inflammation in the intestinal wall and then lead to colitis. So invasive diseases in the case of entamoeba histolytica infections include liver abscess, which is the most common, approximately 1% of symptomatic cases, although in some countries like in Japan, the percentage has reached 17%, and I think that occasionally in some European countries you might also have a liver abscess, which is much higher in some cases, even without the intestinal infections. Astonishingly, very little is known about the immune response underlying the invasive amoebiasis. And previous studies have shown that interleukin-6 was found to have significant levels in some patients that has amoebic liver abscess. Although few studies have also indicated the role of interleukin-10 on the protection of the host against entamoeba histolytica, but those studies were conducted in mice, and no study has been conducted in humans. So the objectives of the study that we conducted were to determine the seroprevalence of entamoeba histolytica, to estimate the occurrence of extraintestinal amoebiasis by the detection of antigen in the circulating blood. Then we also determined the impact of interleukin-10 on the occurrence of IgG among asymptomatic HIV-positive patients. So material and method. So this is our study site. Our study site is situated in the northern part of the country. This is South Africa. Cape Town, most people when they go to South Africa, they visit Cape Town, but we are far in the north. We are somewhere around here. This is Polokwane, and then Louis Trichardt and Tohoyandou is somewhere around here. So this is the Vembe district where the samples were collected, but most of the samples were collected in Polokwane, which is the capital city of Limpopo province. We have Pretoria here, and then Johannesburg down here. So what we did, we collected blood samples from asymptomatic HIV patients. So generally, these are the patients that come to the hospital for a visit, either for their CD4 count or to check the viral load or to collect their medicine. So these patients, they were asymptomatic. They did not have any symptoms. So we use a questionnaire in order to be able to collect some information, like demographic information in terms of origin, age, sex, as well as the CD4 count. And also to know if the patients had other diseases like STDs. So we use the TechLab 2, the entamoeba histolytica test 2, antibody detection, and also the entamoeba histolytica antigen detection. So both these two kits, they came from TechLab from Virginia, sorry. Then we use another kit, ELISA kit, in order to be able to determine the concentration of interleukin-10 among these patients, and the results were analyzed using SPSS. So in terms of the results, this table shows the demographic characteristics of the study participants in terms of the origin, the district where they came from, the area, whether it was rural or urban. We also look at whether the sample was obtained from the hospital, the clinics, as well as the gender, the age group, and the CD4 count level. So the seroprevalence of entamoeba sclerotica was found to be much higher from from the hospitals, from patients who were recruited from the hospitals, and the p-value was significant only at that level. In terms of gender, there was no significant difference, and also for the age group, so it did not matter what the age the patient was, but the prevalence of the seroprevalence of entamoeba sclerotica was quite constant among these different groups. So the samples, when we tested all the 364 samples, only 3 samples were positive for the antigen. So we wanted to look at those samples to see what were the characteristics. So the age varied from 29 to 64, and all the 3 patients were females. And then they came from 2 different districts, and the area where they lived was mostly rural. Their CD4 count was above 200, which is about 350 on average. And in terms of the antibodies, 2 of those samples were positive, while 1 was negative. So in terms of the antimoeba antigen, so they were positive. And for the IL-8, all the 3 of them had a lower concentration of interleukin-10. So in terms of the distribution of IL-10 by age and by gender, so there was no significant difference among the different age group and gender. So in terms of the occurrence of entamoeba sclerotica in relation to the concentration of IL-10, so we can see that those samples that had higher concentration of IL-10, they had much lower prevalence of antibodies, and the p-value was significant, 0.006, which is less than 0.05 that we're targeting. So this is an indication that patients that had produced more IL-10, they were kind of less as risk compared to those that produce less IL-10. So these figures also show similar results. So we can see on the figure on the left, we have the participants with IL-10 more than 2,000 picogram. And then on the figure on the right, we have entamoeba sclerotica seroprevalence. So we can see that those patients that had lower CD4 count, they had more infections compared to those that has lower CD4, that has lower IL-10 concentrations in their blood. So in terms of the conclusions, we demonstrated a high seroprevalence of entamoeba, which shows that this infection is quite common in the area, with about 65% of individuals living in the area having been in contact with the pathogen. Amoebic liver abscesses might be less common because of the prevalence of 0.5%. Elevated levels of IL-10 in patients may serve as protection against the parasite. So we were able to show that the higher the level of IL-10 in the serum, the lower the seropositivity. So the occurrence of entamoeba sclerotica in this study is of public health significance, and if appropriate care is not taken, it could result in epidemic situations. So this study also observed that the infection is more prevalent in rural areas, If you look at other studies in other countries, like in Bangladesh, for example, the prevalence of entamoeba was much higher in townships, compared to the villages. But in this case, we found higher infections in the villages, in the rural areas, compared to town. Yeah, so I would like to thank our funders, the National Research Foundation of South Africa and the Department of Science and Technology, and the patients who participated in the study, and the students who helped with sample collection. Thank you very much.
Okay, thank you very much. So we open the floor for questions, comments, or contributions. Okay, there is one.
Was there any correlation between the amount of IL-10 and the amount of interferon? Did you measure the pro-inflammatory cytokines?
No, we did not measure the other cytokines. So because we went from the hypothesis that was generated by Hamano and his colleague in 2006 at the University of Virginia, who demonstrated using the mice that mice that had high IL-10 tend to be protected. So we wanted to verify that in the humans. So we did not test for other cytokines.
Okay, because IL-10 is often the end product of an inflammatory response, like interferon, TNF, and so you get an inflammatory response in your liver abscess, which then induces IL-10, rather than it's actually protective.
Okay.
I mean, it might be worth looking at the other cytokines to see over time as well how it changes.
Okay, yeah, thank you for the advice.
Any other question?
Yeah, hello. Thank you for a great talk. I just wanted to know what influenced the cutoff for your IL-10 concentrations you used in your study?
Yeah, it was based on the overall distribution. So we tried to find a middle way. So it was not informed by any official data. So we just decided based on the overall distribution of IL-10 in the population. So then we wanted to characterize them, one group that we considered lower and one that we considered higher. Yeah.
Yeah, I'm just asking that because you base on this cutoff to tell this is high and this is low. And then at least based on what the earlier question that was asked, at least if you were able to look at the ratio of this IL-10 to any pro-inflammatory cytokine response, that could have been more informative.
Yes.
Yeah.
Yeah.
Okay. Any other one? So in the absence of any other questions, thank you for the presentation. I'd like to call on Dr. Philemon Kara. Dr. Philemon Kara, who is a social anthropologist, Institute of Robert Koch, Chair UNESCO, the Biotechnique University, Alexandra, Cote d'Ivoire, who is presenting on the preparedness to response, a social anthropological contributions from study of the funeral rites in Cote d'Ivoire. Thank you. Welcome.
Okay. Bonjour à tous et à toutes. Allô. Voilà. Depuis 3 jours, nous avons écouté les sciences biomédicales. Et ce matin, je vais vous parler des questions culturelles et sociales. Donc, ma communication est intitulée de la préparation à la réponse, une contribution socio-anthropologique à partir d'une étude de dérive funéraire en Côte d'Ivoire. Donc c'est pratiquement une dimension appliquée de la communication que j'ai faite le premier jour. Donc, il faut dire que ce travail a commencé en 2015 lors de l'épidémie de la maladie à virus Ebola en Afrique de l'Ouest. Et pendant cette épidémie, nous avons mené une recherche qui portait sur 5 pays de l'Afrique de l'Ouest. Et dans ce programme, en Côte d'Ivoire, j'ai fait mon master. Et pendant cette période, mon travail de master a consisté à faire une analyse du vécu social des mesures qui interdisaient la manipulation des corps en Côte d'Ivoire. Et quand on a fini cette recherche, le 15 mars de 2015, on est allé faire la restitution. Et à la fin, le directeur de l'UNHP, qui est une institution qui est en charge de la mise en œuvre des politiques de réponse en Côte d'Ivoire, a pris la parole et il a dit: si vous devez continuer les recherches sur une seule chose, ça sera sur les rites funéraires. Donc c'est à la suite de ça qu'on a mis en place le programme rituel funéraire et épidémie en Côte d'Ivoire. Et on a mené les recherches parce que cette question, on l'avait en Guinée et au Congo, a été parfois très complexe et délicate. Donc le type de mon travail, c'est de montrer comment la socio-anthropologie peut contribuer à articuler impératifs culturels et non biosécuritaires. Donc pour faire ce travail, nous avons donc décidé de faire une ethnographie des dérives funéraires pour se préparer. Donc on est parti des méthodes classiques de l'anthropologie, telles que l'immersion longue et la recension de documents avec des entretiens approfondis. Donc notre travail a consisté donc à faire une anthropologie des occasions de contact, qui a consisté donc à identifier sur la base du risque construit par la santé publique. Nous avons essayé d'identifier les moments où on touche le corps et les moments où on ne touche pas le corps, et les logiques qui font qu'on touchait ces corps-là. Donc à chaque niveau, nous avons recherché donc ce qui est interdit, ce qui est obligatoire et ce qui est permis dans ces pratiques. Et ce travail a permis donc de prendre la mesure de ce que peut induire l'introduction des normes biosécuritaires, et ensuite d'identifier les contre-rites et les fenêtres d'opportunité pour essayer de comprendre comment adapter les mesures de biosécurité à ces pratiques. Et à l'issue de ce travail, il faut, nous avons tiré quelques leçons. C'est que pour les populations, tout cadavre comporte un risque. Donc ça veut dire que avant même que la santé public n'a communiqué sur les corps comme étant des des objets à risque, ils le savent déjà. Ensuite, nous avons identifié aussi que la dignité, la question de la dignité varie en fait selon les aires culturelles et aussi selon les situations de décès. Et aussi, c'est que cette autre question des dérives funéraires est imbriquée en fait à des logiques de réputation. Nous avons aussi trouvé que toutes les situations de décès font appel à des arrangements dans les enterrements. Et ça veut dire qu'on peut aussi utiliser ces pratiques-là pour adapter les mesures. Et aussi, nous avons trouvé que la variabilité et la diversité des rites funéraires donnent des opportunités d'adaptation, contrairement à l'idée de la complexité. Et aussi, on a trouvé que la méthode de dimension longue ne peut pas toujours être appliquée en situation de crise. Donc, nous avons proposé que pour répondre aux épidémies, il faut déjà considérer le risque funéraire dès les premiers signes administratifs. C'est-à-dire qu'il ne faut pas, comme on a pour habitude de communiquer pour dire que de ne pas manipuler les corps, mais le risque funéraire, ce n'est pas après le décès, mais ça commence dès les premiers signes de la mort. Et aussi, il faut promouvoir une approche systémique intégrée, une approche de la reconstruction. Et il faut aussi réviser les méthodes anthropologiques pour les adapter. Et c'est cette base, à partir de ce que nous avons appris en Côte d'Ivoire, en 2020, j'arrive en France et c'était en pleine période de la première vague de la pandémie. Et l'occasion m'a été offerte d'intégrer un programme de recherche à l'hôpital européen de Marseille. En mars, le 30 mars 2020, j'assiste à leur première réunion de crise et ce jour, la question qui était à l'ordre du jour, c'était la question des rites funéraires. Ils avaient une grande préoccupation parce que c'était interdit et ils ne savaient pas comment prendre en charge cette question. Et donc, je procède à une analyse contextuelle rapide de l'environnement même de l'hôpital. Il se trouve que c'est un hôpital qui est fréquenté par un groupe assez hétérogène, donc les Maghrébins, les Gitans en particulier. Ensuite, c'est que les relations sont parfois tendues, d'où la peur des messieurs par rapport à cette question en cette période de crise. Donc, la question des dérives funéraires, on voit que c'est traversant parce que quand la question s'est posée, comme partout dans les hôpitaux en France, l'hôpital européen de Marseille va sectoriser ces différents services. Donc, le service, il va avoir service Covid et service non Covid. Ensuite, l'équipe mobile va être divisé en deux et il y a une partie qui garde le nom de l'équipe mobile des soins palliatifs et l'autre va être dédié au service Covid. Et le problème, c'est que tous ces services qui étaient divisés étaient restés ouverts à la morgue. Sauf la morgue avait accès. Donc, on a échangé et les premières discussions ont permis de savoir que pour pouvoir prendre en charge cette question, il faut recueillir les informations et les transmettre aux responsables de la morgue afin qu'ils puissent anticiper l'accueil des familles. Pardonnez. Ensuite, nous avons identifié qu'il fallait donc il fallait aussi mettre en place un dispositif de travail collaboratif de sorte à mutualiser les savoirs, parce que habituellement les équipes ils ne travaillent pas avec le responsable de la morgue. Et aussi, on a trouvé qu'il fallait aussi trouver un moyen pour maintenir le lien entre les équipes de soins palliatifs et la suite des événements mortuaires. Donc, suite à ce travail des choses d'analyse contextuelle, nous avons proposé de construire une cellule rite funéraire au sein de l'hôpital et qui avait pour objectif d'encourager les cloisons.
Can you round off in the next 2 minutes? 2 minutes to round off.
Donc, de fini. Ensuite, cette cellule avait pour objectif donc de soutenir toute l'équipe pour qu'ils puissent travailler ensemble. Donc ça, c'est le témoignage du médecin de l'équipe mobile dont j'ai pris sa phrase pour faire le titre. Penser les morts pour penser les funéraires pour penser les morts et réparer les vivants. En gros, c'est que ce travail permet non seulement d'aider les familles, mais aussi de permettre aux équipes de soins de pouvoir se sentir accomplir leur travail. Donc, en conclusion, nous disons que ce travail donc, permet donc de rappeler qu'on peut mener une étude au sud, une étude une perspective au sud et aller l'appliquer. Et cette étude qui se fait dans un contexte qui n'est pas nécessairement un contexte d'épidémie, mais les résultats peuvent être appliqués ailleurs, et surtout de faire la promotion de l'approche de la co-construction, parce que les décisions doivent être prises en commun accord avec les familles des personnes malades, puisque c'est eux qui savent, qui connaissent l'essence de leur pratique et de ce qui peut être digne pour les morts. Je vous remercie.
Okay, thank you very much, Mr. Furman. Before I open the floor, obviously it's an interesting topic, looking at the cultural diversity of Africa and how serious we took issue of parental right. How do you think a sociologist will work with the biomedical scientist to address the issue of I believe we have to take our dead bodies, we have to give them the rights they have, and then not giving them is taboo in some communities. So how do you convince people that in some certain situations, such kind of things might be dangerous to them, looking at the risk in handling especially dead bodies from the pandemic? Thank you.
C'est comment j'ai pensé le risque et
how do you convince let's say traditional rulers, community leaders of knowing the risk, the biosafety risk in taking dead bodies of people who died of infectious disease, especially during pandemic, Ebola, COVID-19, and so on and so forth. It's a very big problem that people traditionally and culturally wants to take their bodies, dead bodies, or give them funeral rights, and so on and so forth. So how do you, how can you, what role can you play in educating people to know that there is boundaries, there is risk in trying to make sure they give the funeral rights to somebody who died of highly pathogenic diseases?
Okay. If I understand you say how I communicate about the risk with the family to accept the measure or
so I open the floor for a question. Question? Comment? Okay, so in the absence of questions, Dr. Freeman, thank you very much.
Thank you.
So I don't know, is the Dr. Maka around? The online? Is he? Okay, so we could not communicate with Dr. Maka Chibo, who should be presenting online. So, and in the absence of his presentation, this brings us to the end of this session. Thank you for your time. Thank you for your attention. Thank you.
Right, thank you very much. Hello? Hola. Hello? Yeah, okay, it's working now. Right, so thank you very much, Dr. Aliu, for chairing that session. We have the next chair in the person of Prof. Shehu Indams. Prof. Shehu, could you please take up? Thank you, sir.
Thank you very much for calling me and giving me the opportunity to chair this. I will call the first presenter in this session, Dr. Aliu. Dr. Aliu is from the Center for Mycotoxin and Food Safety, Federal University of Technology, Minna. He will be presenting on the title, Seroprevalence and Assessment of Knowledge, Attitude, and Practices of Camel Handlers regarding brucellosis and consumption of camel milk and urine in East Senatorial District of Yobe State, Nigeria. You're welcome.
Good afternoon. Distinguished ladies and gentlemen, I'm Harun Aliu from Africa Center of Excellence for Mycotoxin and Food Safety, Federal University of Technology, Minna, Nigeria, to present on seroprevalence and assessment of knowledge, attitude, and practice of camel handlers regarding brucellosis and consumption of camel milk and urine in East Central District of Yobe State, Nigeria. Thank you. Well, of introduction, brucellosis, it has also several names depending on the region. It could also, it's called Mediterranean fever, undulating fever, Malta fever, rocky fever, etc. Brucellosis is an important but neglected zoonosis that cause serious economic losses in livestock and pose a serious public health challenge in human. Infection in animal could occur when animal ingest contaminated feedstuff, when their feedstuff is contaminated with a Brucella organism or through contaminated water. While women acquire this infection mainly through injection of contaminated milk or milk product, or when they come in contact with infected animal or aborted fetal or fetal membrane or discharges from infected animal. Women unpasteurized camel milk previously was mainly consumed by pastoralists, but recently it has gained wider market and commercialization because of its therapeutic effect, and also it's used for prophylaxis in major diseases that are prevalent in Africa, like asthma, diabetes, hypertension, and even in the management of cancer patients. Brucellosis has been reported in many developing countries, like in Nigeria, with up to 13.5% prevalence rate. And this makes it second most important zoonotic disease after rabies. According to OIE, over 500,000 cases of human brucellosis are reported annually. And according to OIE, FAO, and WHO, globally, brucellosis is being underreported. And most clinicians are not familiar with some of these with disease. Even when they are confronted with a patient, it really comes to their mind. It's one of the last diseases that come to their mind. Mostly they engage in treatment of malaria, typhoid, and others. Brucellosis in camel, like in other species, is an insidious infection. That is, it hardly provoke any, you hardly see any clinical manifestation. Camels are believed to be a reservoir of this disease. They only acquire this infection from other small ruminants like cattle and sheep and goat. Human brucellosis is a multisystemic disease with broad spectrum of clinical manifestation with varying degree of organ and system complication. It affects different organs like musculoskeletal system, gastrointestinal system, and even nervous system. Camels are used for various purposes, for the production of milk, wool, transportation, and meat. Diagnosis of brucellosis poses a serious challenge to clinicians and even diagnostic laboratories at the same time. Because it's one of these diseases with protein manifestation, so it mimics other diseases clinically. Objective of this research was to assess the knowledge, attitude, and practice of camel pastoralists towards brucellosis in Yobe State, and also to determine the seroprevalence of brucellosis in camels. And lastly, was to determine the intrinsic and extrinsic factor of age, sex, and location that influence distribution of this disease in camel. Methodology. A cross-sectional study was conducted, and conveniencing sampling technique was adopted to sample camels. And also, a structured pre-tested questionnaire was administered to camel handlers. Methodology that was adopted. Laboratory analysis of serum samples was analyzed using Ross-Benger plate test and serum agglutination test. Data generators were analyzed using SPSS version 20. Descriptive statistics of proportion were used to describe the variables. Also, chi-square and Fisher-Isaac tests were used to test for association. And statistical significance was set at p-value of 0.05. Result. Majority of herdsmen do not have knowledge about camel brucellosis. Some of them know that their brucellosis affect ruminants, but majority of them doesn't have knowledge about brucellosis in camel. Poor practice towards the infection was reported. They keep mixed animals in the same grazing area and even source of water. And majority of them doesn't quarantine their newly purchased animal before adding it to the herd. You can see that about 96% of them throw away carcasses or dead fetus anyhow, and this can serve as source of risk factor in the spread of these diseases. About 100% of them doesn't take any precautionary measure or basic hygienic procedure, that is protective measure, or cleaning of their hand using soap after handling these animals. Table 1 shows the prevalence of Brucella abortus in camel samples using RBT test and serum agglutination test. From this result, more camels were found to be seropositive to Brucella using RBT tests when compared to serum agglutination tests. This is unexpected. From this result, more camels were found to be seropositive to RBT tests when compared to serum agglutination tests. This is an unexpected finding due to the fact that RBT test is a rapid screening test with relatively low specificity but high sensitivity when compared to serum agglutination test. More camels from Gendam local government show more specificity compared to usury. But there was no statistical difference between prevalence rate between the two local governments at p-value. Okay. But there is no statistical significance between the prevalence rate in the both local government for both tests. The difference in the prevalence rate between the local government was not statistically significant for both RBT tests and serum agglutination test as p-value of 0.05. Table 2 shows seroprevalence of B abortus infection in camel based on sex using RBT tests. More females were found to be seropositive for both. More females were found to be seropositive for both tests. This could be due to the fact that females undergo some physiological changes, and also in the female milk there is a presence of erythroid, which make them more susceptible to brucellosis when compared to male. From the result, females show high prevalence rate than male, which was statistical significance. Table 3 shows seroprevalence of B abortus in camel based on age using RBT test and serum agglutination stage. From the result, adult camels were found to be more seropositive for both RBT tests and serum agglutination test. This could be due to the fact that more adults has been in the environment, and considering the management system where extensive system is being practiced. And according to Kaltungo et al. In 2018, he said management system is one of the factors that aid in the spread of brucellosis among herds, because here the farmers keep both small ruminants and then the cattle, and camel, as we know, they are reservoir, so they acquire this infection from other animals through spillover. Table 4 shows risk factor associated with camel brucellosis. Age and sex were identified as proportional risk factor for potential risk factor for camel brucellosis and were estimated using odd ratio. Results show that females had 0.57 more chances of getting infected than male. This association was found to be statistically significant. Also, age, the results showed that the adult camel were at higher risk of infection with odd ratio value of 0.07. A statistical significant association was observed for this finding. Conclusion. Majority of herdsmen do not have knowledge about camel brucellosis in the study area. poor practice towards the infection were reported. Again, seroprevalence of Brucella abortus infection in the survey camels were 42.5% using RBT tests and 22.4% with serum agglutination tests. Seroprevalence rate was higher in Gendem local government and lowest in Unisari local government. Female camel had significant high seroprevalence and adult camel had significant high seroprevalence. Recommendation: the government should institute a national policy, routine surveillance and control program for brucellosis in livestock, including camel, to check the spread of disease in the country. Further studies should be considered to ascertain the presence of B. abortus antibody antigen in the milk of camel to ensure wholesome milk for the populace. Public health campaigns should be instituted to increase awareness of brucellosis and an appropriate preventive measure against the disease. I wish to acknowledge the SA partner, WANIDA Symposium team, African Center of Excellence for Mycotoxin and Food Safety, Federal University of Technology. I wish to acknowledge my center leader, Professor Hussein Makun, and his team, and my supervisory teams. Thank you for listening.
Thank you for that timely presentation. The floor is now open for questions. Yes, please. Behind. Okay.
Thank you, Ali, for that presentation. The one thing that got me really interested in the work is where we talk about the urine and its uses, that the urine of the camel. Yes. I don't have any information on that, and I guess it's something I would wish you to shed more light on as to, you talked about its, is it therapeutic use? And I guess that's why you decided to also look at it in terms of the seroprevalence of the disease. So please, can you shed more light on that? Yeah. Thank you.
Thank you for that information, for that question. Even over 500 years ago, in Asia and Africa, camel urine has been used for different therapeutic purposes and then management of certain diseases. Before now, we are thinking probably because we are from Africa, where healthcare system, modern healthcare system, is not reachable for majority of the population, and where traditional medicine is in increase. The users of patronage of traditional medicine is in increase. But you will be surprised that even Saudi Arabia Peninsula, a lot of the patients, especially those that are suffering from female breast cancer, patronize a lot of this camel urine. Even for prophylaxis, even if you are healthy, they use it. And scientifically, there has been a lot of work to find out what is the content, what is there in the urine. And they realize it's contain cytotoxic. There is a substance in the urine that have cytotoxic effect, which can be utilized even from to prevent metastasis of cancer cells. It impaired development of cancer cells. In the north, it's a popular, I have seen families that use this urine for different purposes. Because once you are sick, you want to get solution anyhow. People, the issue of WHO was warning when the Middle East respiratory outbreak happened in Saudi Arabia, that it's possible, if people are not careful, they could contract this infection through the drink of this camel urine. It's on sale. There are regions, when you go, places like Katsina, it's sell like they sell petroleum in the black market. They will say this one is not, is pure, this one is diluted, and that's how they fix their price.
Okay, I'm also thinking, if that's the case, is it not possible that the urine, as it were, may also be inhibiting the brucellosis?
No.
That is the brucella?
Yes.
As in, will not be also toxic to the organism?
I'm coming. Brucella militensis from small ruminants. It's the most pathogenic of the all species of Brucella, and it has been isolated from camel urine. It impairs other Brucella organisms, but it doesn't really have effect on Brucella melitensis, which is most pathogenic of all the species. Thank you.
Okay, do we have more questions? Yes, please.
Thank you very much. And thank you for the presentation. Is it human brucellosis endemic in your country? And did you do diagnosis for Brucella abortus in human infection cases?
Yeah, SANI 2019 report from his study, he report high prevalence of Brucella and seroprevalence in sample in his study in Niger State. Even though his study targeted on high-risk individuals, butchers, veterinary practitioners, and then pastoralists, he came out with high prevalence in human, which shows that this disease is going on in the population. And unfortunately, even the medical practitioners are not too familiar with the clinical manifestation of this disease. So anytime patients are confronted with them, is one of the last things that come to their mind. It rarely comes to their mind to be put in part of their diagnostic procedure. Again, brucellosis has been found to cause death in human because acute chronic cases that are not managed can result into endocarditis, which eventually can lead to death. And also, it has been reported to cause arthritis and several organs and system complication.
Okay, thank you for that response. I'll call for one more question, then we end this session. Do we have more questions? Okay, thank you for a nice presentation. Can we clap for him? The next presenter is Languilla, who is a PhD student in the University of Guiba in the Republic of Congo. The topic is SARS-CoV-2 infection in pests from COVID-19 positive households in Brazzaville, Republic of Congo. It's an online presentation. Are you available? Okay. Please present your screen and then present. Thank you. Hello, Hussein? Hello, we can't hear you. Sorry?
We can see on screen maybe.
We can see your screen. Can you go ahead with the presentation? We can't hear you. We can't hear you. Can you unmute your mic? ICT, can you assist us to get to her? We'll get back to you later after the next presenter, please. She's already gone.
Is Ibrahim, please, can you come?
Thank you very much. Good afternoon. Karid Jori Ibrahim from Sermes, and my today's a valuable means for the prevention or proper communicable disease in infants and children. Nutrition is, as you all know, one of the largest single worldwide malnutrition, as you all know, is one of the worldwide largest single underlying cause of death, child death, especially in low and middle income countries, as the case is in South Asia and most of the sub-Saharan African countries. So malnutrition kind of promotes the incidence, chronicity, severity, and case fatalities of commonly encountered infectious diseases by compromising the immune system and making the infant more susceptible to these infectious diseases. There's a kind of synergistically and bidirectionally linked relationship between malnutrition and infectious diseases that make them a very huge public health problem, especially in countries like mine. So, usually the most common form of this malnutrition, that's the moderate form, is not a life-threatening condition, but it's a threefold risk factor to the life-threatening condition, that's the severe form of the malnutrition, and the one that is mostly associated with infectious diseases. So what has been also been noticed with this kind of malnutrition is both kinds are associated with dysbiosis, because usually they occur in at a period where the child and the infant are seeing the establishment and maturation of their microbiota. And this malnutrition would create a condition that would kind of perturb the proper establishment of the microbiota, meaning that when you treat infants with the commonly used treatment, that's therapeutic flowers, they help, these standard therapeutic flowers help the refeeding process, but the infant remains stunted and at risk of undergoing subsequent episodes of malnutrition because of the dysbiosis prevails. So what currently is feeding scientific literature is that when you want to address malnutrition, that's the best way to go through is to also, as you are trying the refeeding process, also target the microbiota for its amelioration and its maturity. So that's what we are trying to do. It's obvious that the nutritional status of the infant has a crucial role in the prevalence, incidence prevalence and the outcome of this malnutrition. Surely the associated infectious diseases. So we are trying in a One Health mindset, you are trying to address this malnutrition by producing local made with local ingredients, therapeutic flowers that are rich in prebiotics, especially prebiotics that are of polysaccharide source. So the way we go through it is through this project. We still have a long way to go, but at least we've started with this project co-funded by the AS and CE partners. So the way we are going through it is first we've looked up into the dishes of mothers, caregivers, and advisors for the prevention of this malnutrition in three regions of Niger, a region that has a high prevalence of malnutrition, a region that has a low prevalence, and a region that has intermediate prevalence of malnutrition. So what we've been able to gather is what we are doing in the next step, look to assess for the biochemical content and also the prebiotic potentials of these prebiotics that we shall use in the work package 3 in a formulation of therapeutic flowers that we shall use then in a clinical trial while monitoring the clinical status, nutritional status alongside with the eventual impact these therapeutic flowers would have on the microbiota. So this is a very recurrent and quasi-permanent health issue that we have in our context and that we are trying to address with a One Health approach through addressing the malnutrition aspect because of when you primarily address malnutrition is like you are addressing, you are lowering a risk factor to the infectious diseases that are associated with the complicated, that's the severe form of malnutrition. So this is just an introduction of a new research area that we've started in our lab that could come and complement what has already been explained by my scientific director this morning. Thank you very much.
Yeah, thank you, Ibrahim, for the presentation. Are there comments or contributions to what he has presented? I'm sure it's an interesting area and it calls for interactions. So now that here you can, okay, there's somebody here. Thank you.
Thank you so much for the presentation. In terms of malnutrition, I don't know if there's any project that has been done looking at the impact of intestinal inflammation and the gut microbiome in the area where you are. Second question is concerning the medicinal plants. Generally, when we were growing up, there are certain plants that are given to children when they are not eating well. I don't know if there has been any investigation, to see if malnutrition can be improved with the application of such plants.
Okay, to your first question, I would say that there are good literature reports concerned with the inflammation and microbial translocation that is favored during a dysbiosis that causes this malnutrition. And with respect to the traditional plants that you are talking about, is because of that in our first work package, we've been interested in a socio-anthropological survey to look into the dishes of the indigenous, a kind of ask for the traditional way they used to handle or to prevent this malnutrition. Is a way for us to say, since those people are not having such problem and you are in the same area, so how is it that they are not having the problem? Is it the way they are feeding their children with the same thing that you could have available to you? So if that is the case, so let us have a scientific basis of the usually done traditional process for this prevention and management of malnutrition, and come back to the mothers, caregivers, grandmothers, or advisors, to come back to them with a standard formulation of this flowers that could be homemade, so that everybody in every area in Niger and at any time could have these ingredients and have a homemade of these flowers that could help in the prevention and management of the malnutrition, and by the way help reduce the incidence of infectious disease that could be eventually associated with the severe forms.
Do we have more comments or questions? Okay, can we clap for Ibrahim, please?
Thank you very much.
Are we ready with Hosanna now? It's like you've made yourself up.
Bonjour tout le monde, est-ce que vous arrivez à m'écouter?
Yes, we can hear you.
Ok. Bonjour tout le monde, je suis Osana Yendia, doctorante à la faculté des sciences et techniques de l'université Marie-Ngouabi. Je vais vous présenter la synthèse de mon travail de thèse qui porte sur l'infection à SARS-CoV-2 chez les animaux de compagnie vivant avec des propriétaires testés positifs à la COVID-19 à Brazzaville en République du Congo. Ma présentation comprendra 4 parties. En effet, depuis le début de la pandémie de COVID-19, plusieurs cas des animaux testés positifs au SARS-CoV-2 sont rapportés en Asie, en Europe et en Amérique. Au niveau de l'Afrique, une seule donnée rapportant l'infection d'un puma et 2 lions a été rapportée en Afrique du Sud. Cependant, la grande majorité des animaux infectés par le SARS-CoV-2 sont des animaux domestiques. Au niveau de la région africaine, selon notre connaissance, il n'existe aucune donnée rapportant les cas d'infection des animaux de compagnie par le SARS-CoV-2. En effet, nous sommes sans ignorer que, au niveau de l'Afrique en général et au Congo en particulier, il existe un grand nombre des animaux de compagnie et les différentes interactions qui existent entre l'homme et les animaux ne nous permettent pas d'exclure le risque d'infection de ces animaux par le SARS-CoV-2. C'est alors dans ce contexte que nous avons émis comme hypothèse que le SARS-CoV-2 circulant dans la population humaine pourrait être transféré aux animaux de compagnie et ces derniers pourraient servir de relais entre la population humaine et les animaux sauvages environnants. Pour vérifier ces hypothèses, nos objectifs ont été d'investiguer les cas du SARS-CoV-2 chez les animaux de compagnie, de caractériser les différents variants infectant les animaux de compagnie, ainsi que de déterminer la séroprévalence de l'infection à SARS-CoV-2 chez les animaux de compagnie à Brazzaville. Pour atteindre ces objectifs, nous avons mené de février en décembre 2021 cette étude en se servant de la base de données nationale du Congo concernant la COVID-19. Nous avons pu recenser les patients testés positifs à la COVID que nous avons contactés pour avoir leur consentement. Après le consentement des propriétaires, nous avons pu inclure un certain nombre d'animaux dans cette étude en réalisant des prélèvements ainsi qu'en collectant des données socio-épidémiologiques et des données cliniques. S'agissant des prélèvements des animaux, nous avons réalisé en premier lieu des prélèvements des écouvillons nasopharyngés et rectaux en partant du principe que, une fois le propriétaire des animaux testé positif au COVID-19, dans un intervalle de moins de 15 jours, nous l'avons contacté pour prélever son animal de compagnie. L'animal testé positif au premier prélèvement a été reprélevé chaque 2 jours jusqu'à ce que son résultat s'est avéré négatif. Les animaux testés négatifs au premier prélèvement ont été reprélevés 2 jours après pour confirmer le résultat. Vers la fin de cette étude, nous avons réalisé des prélèvements sanguins chez les animaux testés positifs et négatifs aux écouvillons nasaux et rectaux. Comme technique de laboratoire, nous avons utilisé la PCR en temps réel en ciblant 3 régions du SARS-CoV-2, à savoir une région des polyprotéines ERF1AB, la protéine S ainsi que la protéine N. Les échantillons testés positifs ont été analysés en utilisant le système MySeq et les échantillons sérologiques ont été analysés en utilisant la plateforme Luminex afin de rechercher 3 types d'anticorps dirigés contre le SARS-CoV-2, à savoir les anticorps anti-N, anti-S et les anticorps anti-RBD. Comme résultat, sur les 10 mois qu'a duré cette étude, environ 6000 personnes ont été testées positives au SARS-CoV-2 au niveau du Congo, parmi lesquelles nous avons pu contacter 5000 personnes. 185 ont confirmé avoir au moins un animal de compagnie chez eux et 65 nous ont donné l'autorisation pour pouvoir prélever leurs animaux de compagnie. Dans l'ensemble, 100 animaux ont été prélevés pour les analyses de biologie moléculaire et 65 ont été prélevés pour les analyses sérologiques. S'agissant des prévalences de détection du SARS-CoV-2 chez les animaux de compagnie, sur 100 animaux prélevés, 14 ont été testés positifs en PCR et 22 en test antigénique. Donc, dans l'ensemble, les prévalences décrites chez les animaux de compagnie au niveau mondial sont assez variables. On note les prévalences allant de 5,3 à 33,3 concernant la détection de l'ARN et les prévalences de 7,8 à 44,7 concernant la séroprévalence. S'agissant du suivi de l'infection des animaux de compagnie, 37% des animaux ont présenté des signes cliniques liés au SARS-CoV-2 comme on observait chez les humains, à savoir les éternuements ainsi que les difficultés respiratoires. D'autres signes cliniques, comme les signes cliniques relatifs aux gastro-entérites, ont été également notés chez les animaux. Les animaux ont excrété le virus pendant une durée allant de 1 à 12 jours pour les chiens et de 1 à 17 jours pour les chats. Quant aux différents variants détectés chez le SARS-CoV-2 chez les animaux, sur 14 animaux testés positifs par PCR, 10 ont pu être séquencés et les séquences obtenues correspondaient aux variants bêta, aux variants localement appelés congolais, ainsi qu'un variant recombinant entre les deux variants précédemment cités. Les variants décrits chez les animaux correspondaient aux variants ayant circulé pendant la période de novembre 2021 en février 2022 dans la population congolaise ainsi que dans la population des pays voisins. Pour conclure cette étude, nous pouvons dire que l'infection à SARS-CoV-2 peut bel et bien avoir lieu en Afrique ainsi qu'au Congo. Et les différents variants détectés dans cette étude ont aussi été détectés chez la population humaine. On peut dire que les animaux peuvent excréter le virus pendant une durée de plus d'une semaine. Sur ce, me voici à la fin de cette présentation. Je remercie toute l'équipe d'organisation de WANIDA pour l'opportunité qui m'a été accordée de présenter ces données et je vous remercie pour votre attention.
Thank you very much for that presentation. The floor is now open for comments, questions. Are there questions, please? Okay.
Thank you for the presentation. I'm interested in knowing if the type of COVID variant that is found in the animals, if it's the type that can be transferred to human. I'm not sure I got that during the presentation. Thank you.
Merci beaucoup pour votre question. Pour essayer de répondre à cette thématique, à ce jour, il existe seulement 2 cas de transmission inverse des animaux de compagnie à l'homme qui ont été rapportés. Mais notre étude ne concernait que la partie de transmission de l'homme aux animaux de compagnie. Nous n'avons pas pu analyser le volet de transmission inverse des animaux de compagnie à l'homme.
All right, thank you.
Do we have more questions? Yes, please.
Merci pour votre très brillante et intéressante présentation. Le sujet m'intéresse très bien. En fait, la question, peut-être je vais poser autrement la question de notre collègue, est celle de savoir si les variants ou les génotypes, je ne sais pas, que vous avez trouvés chez les humains, est-ce que c'est les mêmes que vous avez eu chez les animaux infectés? C'est la première question. Des deux, comment pouvez-vous prouver que ce n'est pas plutôt la transmission inverse qui a eu lieu? Est-ce qu'au moment où vous avez prélevé l'animal, comment savez-vous que ce n'est pas lui qui a été infecté en premier?
Merci beaucoup pour vos questions, messieurs. Pour essayer de répondre à la première question, je peux dire que comme je l'ai dit j'ai essayé de le dire dans ma présentation, les variants détectés chez les animaux de compagnie ont aussi été détectés chez les humains dans la même période de notre étude. Et s'agissant de la deuxième question, concernant entre l'homme et l'animal qui a été le premier à être infecté, pour la plupart des enquêtes que nous avons eu à effectuer, les humains ont rapporté avoir commencé à développer des signes cliniques de l'infection bien avant leurs animaux de compagnie. Donc sur cette base-là, sans aller plus loin, nous pouvons dire que l'infection s'est faite plutôt des humains aux animaux de compagnie. Mais étant donné que nous n'avons pas eu à analyser dans le cadre de cette étude les animaux, les échantillons des humains, nous ne pouvons pas affirmer de façon très objective que les animaux infectés ont bel et bien été infectés par leurs propriétaires ou par des voisins. Merci.
Okay, thank you for that response, Hosanna. Do we still have more questions for her? If there's none, then we clap for Hosanna, please. And with this, I've come to the end of my chairmanship, and I hand you back to the organizers. Thank you.
Right, thank you very much, Prof, for chairing this section for us. So I think we are all tired, we need to stretch, and it's time to eat. So we'll go for lunch, and we'll come back for our next session. So we have 1 hour break, and we all head towards the restaurant, and then we can have something to eat. So enjoy your meal. Bon appétit. Right.